Synthetic biology and biotechnology are providing tremendous leaps forward in scientific solutions for current global problems. Biofuel production with organisms that create a variety of fuels from renewable feedstocks that replace fossil fuels. New materials such as spider silk, leather replacements, plastic replacements. There's medical applications such as the artemisinin, therapeutic biosensors, targeted drug delivery and gene therapies. There's other products which remedy supply shortfalls or eliminate polluting production processes for the supply of fragrances, flavours, inks and dyes. Also environmental bioremediation organisms which detect pollutants or clean up those plastics and toxic wastes. Now, under our control, these deeply engineered organisms do amazing things, but what if they escape? Wah wah. They could take over our world and change it forever. The ecology of natural organisms could be disrupted, and our amazing synthetic organisms could wreak havoc functioning out of context. Biocontainment is not just about physical containment. It's about being able to prevent the survival of these deeply engineered organisms outside of labs and biotech fermentation tanks. We need to consider not only the survival of the organism, but the genetic material inside them. Not only could the organisms potentially outcompete those in nature, disrupting ecology, but horizontal gene transfer may alter natural organisms. But don't panic. Synthetic biology has the answers. There's trophic answers and semantic answers. So trophic relates to metabolites and controlling organism survival through dependence on an input which is not readily available in nature. Semantic involves changing of codes and mechanisms so that nature cannot interpret the genetic material, which prevents genes from leaking into nature. There's a double benefit with this because it also protects the biocontainment itself by preventing the organism from acquiring a genetic solution from nature. Now, under trophic, there's basically passive and active methods. So passive methods include things like inducible expression, like you've got a lac operon and you don't express anything unless you throw in some IPTG. Or it could be oxytrophy, where a gene is knocked out for an essential metabolite such as thymine and the organism is just dependent on being artificially supplied. Or something called slide, where the ligand is benzothiazole and that's needed to be able to trigger the expression of essential genes. Now, another example is where the host organism was altered to make it dependent on phosphite and hyperphosphite for its phosphorus by adding a phosphite hydrogenase and also replacing its transporter so that it could only allow phosphite. Not only does this create biocontainment because phosphite's so scarce in nature, but it also meant you could select against natural organisms by just having a phosphate-free media. Now, some active types include kill switches that involve complex logic circuits requiring specific combinations of input substances for survival, and they've got cool names like dead man, passcode. For example, an input could affect a promoter for expression of a second repressor, which either represses expression of a toxic substance or it enables expression of an essential gene. Another type is addiction modules. Now, this is where you've got a toxin-antitoxin combination system, where a toxic gene is on the plasmid and an antitoxin gene is on the genome of the organism. Now, this prevents horizontal transfer of the plasmid, which also carries the code for your cool biotech product or function. So if that plasmid is transferred to a new host, that host is destroyed. The most effective of these solutions are multifactorial, things like GeneGuard, where the host may oxytrophically dependent on the plasmid plus the plasmid carrying toxicity for a new host. Or another multi-layer system, which combined riboregulation requiring inputs to translate essential genes, also with toxin, antitoxin, addiction modules, a little bit of oxytrophy and repressor supplementation. They achieved a low 2 × 10⁻12 escape mutants. Now, some question this, saying the detection limit is only 10⁻11, and that's still well above true containment, while others argue 10⁻8 is sufficient. I mean, come on, imagine. That is only 100 colony-forming units in a 1-litre culture, which can easily sustain 10 billion colony-forming units. There probably isn't a one-size-fits-all, as some synthetic organisms and their genes will obviously pose a greater threat if released into the environment than others. But we do want to find a way to reduce those numbers. Now, semantic methods. This is where synthetic biology alters the host and the plasmids to create entirely new mechanisms of that DNA to RNA to protein central dogma, which nature simply does not recognize. The advantage of semantic biocontainment is that it additionally protects the biocontainment method itself by preventing the organism from acquiring a genetic solution from nature through horizontal gene transfer. So, how is this done? One way is to rewrite the DNA code. Some use the amber stop TAG codon and reassign it to a newly synthesized tRNA, which doesn't occur in nature. So while it's within the synthetic organism, the desired proteins are being synthesized. But if that DNA leaks out into nature, it's going to be read with a stop codon. There will be no protein. When these reassignments are inserted into an essential gene, the genetically engineered host can't survive without a supply of the unnatural tRNA. You can add an extra layer to this semantic biocontainment with non-canonical or artificial amino acids. This is when we move into xenobiology. Now, xenobiology has even come up with xenonucleic acid. ZNA, unlike DNA, simply can't be read by a natural polymerase. So the ZNA polymerase and ZNA-directed RNA polymerase and other components required for replication could all exist on their own episomes separate from the host genome, and the episome maintained only by artificial supply of xenobiotic nucleoside triphosphates. Now, with several layers of biocontainment, we can confound any chance of escape mutants. The sustained growth of the bioeconomy, synthetic biology and biotechnology and all the solutions they offer to global issues, it's imperative that we ensure biocontainment, and I believe the scientific field of synthetic biology holds the solutions.